So if you can share your screen.
Yeah, this is a talk by Carlos Santibanez-Lopez, co-authors Jesus Ballesteros, Caitlin Baker, Yoram Zvik, Efreet Gavesh Regev, I hope I pronounced it, and Prasant Sharma. Phylogenetic analysis of 100 scorpion venom gland transcriptomes reveals the origin of toxicity, and they're all from the University of Wisconsin-Madison. So take it away.
Hello, everyone. Can you hear me? Can you see the screen correctly?
Yes.
Okay, perfect. Thank you. Well, thank you so much for coming to this talk. Today, I'm just going to talk about, I'm going to continue the line that Jeff was talking about, venom. So without any further, I just want to mention that venom is just a complex mixture of bioactive compounds that animals, in this case, use to disrupt biochemical and physiological processes in target organisms. It could be either a prey or it could be also to deter predators. As we have seen, venom has evolved independently several times all over the animal kingdom, from sea anemones to mollusks, arthropods, of course, all the way to vertebrates such as snakes. And so within the chelicerae group, our favorite group, of course, this is the latest topology of arachnids published recently by Andrew Antano and his team this year. And we can see that now Scorpionis is actually a sister group to Pseudoscorpionis in a clade known as Panscorpionis. But the reason of this slide is that I just want to draw your attention to the fact that there are 4 major groups of arachnids that possess venom. Including ticks, obviously spiders, as Jeff has talked about before, pseudoscorpions. This is a very cool, interesting group of organisms as well. And finally, the reason of this talk is Scorpionis. And since this topology recently published, it suggests that maybe the origin of some of these venom components in pseudoscorpions, scorpions, and spiders may have originated at the base of Arachnopulmonata, which now includes Panscorpionis and Tetrapulmonata, right? With a subsequent loss of these toxic components in pedipalpi. Okay, but let's now take our attention to the reason of this talk. Scorpions. Scorpions, I guess they don't need to be introduced. There are around 2,500 species that are distributed all over the world except Antarctica, and you can find them at high altitudes, mountains, all the way to the deep caves, going from desert, really hot desert environments to rainforests. We can actually know that there is a division nowadays recognized that just falls into 2 borders, Butida and Yurida, and I'll speak about that in the following slides. But first, I just need to tell you that what has been done about the venom composition in these animals. In the latest years, perhaps since 2000, the vast majority of research has been conducted exploring the composition of those highly toxic scorpions in the family Butidae, right? Those animals that represent a danger to humans. And from those animals, we know that there is a great composition of several things known as toxins, which are small peptides that can adopt basically the cysteine stabilized alpha beta fold here in scorpions, unlike in spiders, like Jeff was telling us. ICK. ICK is not it's not a common type of fold that you will find in scorpion toxins. Now, among those toxins, all the scorpions possess toxins that are insect specific. That means that those toxins are specific to insect tissue, right? But in contrast, those scorpions that are highly toxic to mammals, including humans, we notice that they also have toxins that are really, really specific. And we think, or our initial hypothesis was that since mammals are among the most important predators of scorpions, this must be a kind of defensive counterpart from scorpions to against mammals. So this is one of the things that we want to prove in this talk. And on the opposite side, we can see that nowadays, one of the things that I've been doing in the past 6 years, I've been studying the venom composition of those poor neglected otter families. I forgot to mention, I'm sorry, that scorpions also are, well, the systematics of scorpions nowadays recognize 22 families. And of course, like I said, buthids takes pretty much all the attention in the last 20 years. And nowadays, what I've been trying to do is just to explore the venom composition of those other families that probably nobody has seen because they are not as important as buthids. And so we can, what we can see from these slides is that the distribution of those major components is slightly different. Now, I need to tell you that the category in red, which is indicating toxins, it doesn't necessarily mean that these toxins are with high affinity to mammal tissue. It also can include those toxins that are specific tissue. Sorry, with affinity to specific tissues. Okay, but one of the things that were banning the evolutionary study of venom in scorpions was the fact that we didn't have organized phylogenetic framework until 2015, where Prashant and collaborators published this NICE 3. Although, unfortunately, the sampling was, well, slightly poor. Sorry to say that, Prashant. But there were some organisms, some scorpion species that were missing in this tree. And one of the things that dragged our attention from this initial analysis was that the fact that there were some conflicts with the morphological signal that was recovered in cladistic analysis published previous to this paper on 2015. So the first thing that we did, I was trying to assess the phylogenetic systematic of these animals and try to solve all those weird conflicts that Prashant and his team found out in 2015, right? So the first thing that we did is just try to reinstate the superfamily Behoboidea, right? Because in that first analysis, this family was recovered as polyphyletic. So this study was published in 2019. And then from this analysis, we recovered something also that was quite interesting, the phylogenetic position of Urotonus, longstanding Bijovid, recognized Bijovid. It was not part of that superfamily. In fact, its position was, we needed to do to conduct additional analysis. And so that's what we did in the following years when we were trying to also try to understand what was going on with the phylogenetic placement of the giant scorpions, Hadurus. And now in 2019, we published these two papers about that. We reinstate the validity of the superfamilies Caraboctomnea, sorry, and Aduroidea. And now we're just moving the puzzles to try to fit their corresponding place based on phylogenetic signal. However, one of the things that we're missing here was that we were not sampling enough buthids, right? So we always granted that buthids were monophyletic and there was nothing else to do there because, of course, they share a really simple synaptic morphology. But of course, in these buthids, also we can find all those highly toxic scorpions. And so here, today, I want to just talk about these two questions, unfortunately, due to the time that I have. And the first one is just to see when these highly toxic scorpions diverge. And for that, we sampled more than 100, well, actually 100 transcriptomes. We just removed some of them for the final analysis and the addition of the one genome, Centroretis cultivatus. Unfortunately, the genome of Mesobutus martensi is just, we have seen that it produces a long branch and we therefore just try to remove it. I'm not going to stay here in the method slide, but I'm also happy to take some questions if you want to. But essentially, we're just using orthologs that have been recovered in previous analysis, such as Ballesteros and Sharma. And of course, this is something that Chewy has helped me a lot, from which I'm very thankful. Okay, so this is the topology that we recovered. Again, we recovered essentially the division that Sharma and collaborators presented in 2015. Both part borders were recovered monophyletic. Butida, which comprises three families, Carylidae, Pseudocactidae, and Butide. And then the other one, which is the Urida, it's represented by the rest of the colors, which includes the other 19 families. Unfortunately, we still have some gaps to fill out in the Uridae, but like I said, we have been addressing and we're continuing to addressing these issues in the future. But now let's just focus on Butide, because of course Butide comprises nearly 47%, sorry, of the extant scorpion diversity, and more importantly, it also contains all those, most of them, just a couple of species are not included there, but most of the highly toxic scorpions to mammals are included in this family. So this is the topology, and what is interesting to note here is that on 2006, Fett and collaborators presented a cladistic analysis using trichoboreal characters, and here we were able to recover that topology with some minor differences, especially in the compositions of some of these groups. But the reason of this slide is just to tell you that the groups that contains all those highly toxic scorpions to mammals are recovered independently, not in a monophyletic group, which suggests that this the origin of these animals is not unique, and we have 5 independent origins for these animals in the beauty scorpion tree of life. So applying dating methodologies, we were able to recover this topology. And this is interesting because, of course, in previous hypotheses suggested that beauties were really, really old, perhaps originated back to the Jurassic, sorry. But now here we're seeing that it is actually divergent in the very late of the Cretaceous, right? So here we have a closer look of those red branches, which are all those major highly toxic scorpions to mammals. And here we have those standard deviation of the divergence times. But if we compare this against the origin or the hypothesis of the mammal predators of scorpions, we can see that the origin of these highly toxic scorpions correlates to the origin of their predators, which is just suggesting us that there might be a relationship there between predator and the origin of these highly toxic components in scorpion venom. Okay, so with that, we have evidence that supports the possibility of a relationship between predator and prey. And now let's focus our attention if we can finally, if we can find a single origin of these toxins with these high affinity to mammals. And for that, what we did is just sample all of these transcriptomes and all the data sets, and we retrieved all these venom components, and we have perhaps a better framework to compare the composition of urids and butids. And I'm just going to highlight here 2 things. Calcines are unique to urids, and the butids, some of them, especially those from the old world, will only have the chlorotoxins, those toxins that affect chloride channels. But more importantly, the number of transcripts that we found for the sodium toxin, is greater in the butids. Okay. Now, using maximum likelihood trees and ancestral state reconstruction, we were able to recover the several independent putative origins for these toxins that are with high affinity to mammals in the butid lineage. And here you see that there are 5 major groups for these toxins. I'm not going to mention this, but essentially they are named based on the more representative group. And of course, we based this decision using functional data that has been published through all these years, especially for those scorpions such as Ligurus, Androctonus, and Centroroidus. So those red dots on your screen are indicating the origin of these toxins with affinity to mammals, right? And of course, we can observe that there might be a possible bias due to the length of these amino acid peptides, right? They are very, very short. So we decided to explore other tools such as the clustering methods like CLANS. And here we can also see that by similarity, these peptides with high affinity to mammals are also recovered several times independently from each other, which supports this hypothesis. And more importantly, since we recovered these different groupings of these toxins all over the place, we were trying to see if there were like a signature that may correlate with convergence of these different toxins. And for that, we use the software meme. Please do not confuse. This is not the same meme as Jeff was telling us about. This means just search for these patterns of these consistent patterns in different peptides. And so on your screen, you'll see that the only thing that is unique to these toxins is just the cysteine pattern. And of course, let me just remind you that this pattern is completely different from what Jeff was presenting us with the ICK folding. This is the cysteine stabilized alpha beta. Okay, so here we see that there is no consistent pattern of molecular signatures. So this means that all these toxins have been doing its own thing independently from each other. And of course, this might be also correlated to the fact that we have mutates spread all over the world. And then lastly, I just want to tell you that this also fits into the model that we have evidence, functional evidence, that there is an intermediate state in which a couple of toxins have been discovered in the venom of Leyurus that has this dual affinity that can also affect mammal at the same time that it can affect insect. So our hypothesis suggests that we have the hypothetical housekeeping gene that evolved to become a toxin, the channel toxin, a sodium channel toxin that affects insects or other arthropods, essentially just like the weapon to become a predator. And then that protein was also co-opted to become a defensive trait against the mammal predators. Okay, and with that, I just want to acknowledge a lot of people, a lot of my colleagues in different parts of the world, which would, of course, without their help, this wouldn't be possible. And so if you have questions, if you still have some time to questions, I will be happy to take them.
We're actually running a little late, Carlos, but there was a interesting question from Greta Binford. How confident are you that these are secreted in the venom?
That's a great question, Greta. And of course, all of these transcriptomes were extracted from the venom glands. And of course, we cure them by just doing blast and phylogenomic trees using known peptides with the known function. So essentially, we are pretty confident that these are indeed toxins, and it fulfills also some of the things that Jeff was telling us, like it has the signal peptide, it has the mature peptide, and in some cases also the protectide and all those things.
Yeah.